Yes, ma'am.
Okay, perfect. So, good evening, everybody. My name is Itunuoluwa. I'm from the Department of Computer and Information Science. First of all, I want to thank the organizers of this very fantastic event for inviting me. Thank you for considering me for this. I'm going to just go into the lecture. Because I know it's a tutorial, ideally I would have loved to take questions, but because of time, let me finish. And then at the end of the talk, if you have questions, I'll be happy to answer them. So what you could do is to just take note of the slide number, and during the question and answer time, I'll be happy to go over any part that is not clear. So the topic before me today is a topic that I'm very happy to talk about. It's something that I've been doing. If I'm not mistaken now, I've been actively doing machine learning for bioinformatics. Maybe let me say 10 years. So I'm just going to share with you some of my own ideas and some of the application areas that I have seen and things that I know that we can easily do, and also sort of apply in our own context and be able to bring out new ideas. So this is more or less just me sharing ideas. It has nothing to do with my own personal research. So I'm not bringing my personal research here. I'm just showing you what I know that we can do. I've also implemented some of them before, but I'm happy to just give you like a broad overview of how our expertise in artificial intelligence can be useful in biomedical research. So this is the outline of my presentation. As a form of introduction, I'll just talk about bioinformatics generally. And so bioinformatics, we know that it's an interdisciplinary field. As the name implies, you're seeing informatics, you are seeing bio, and that bio, it's a very big word, because when you say biology, it involves a lot of things. So there's microbiology, there's biochemistry, there's biotechnology, there's molecular biology. There are all types of biological sciences that have been woven together. And then the informatics part is also there. So the word interdisciplinary is a word that I don't want you to forget. But basically what we are doing is that we are just trying to apply our knowledge and our understanding of computational techniques to be able to analyze and to interpret biological data. So if you look at this picture to my right, you would see that there's a mix of a lot of things. So first of all, you have computer science, you have chemistry, you have engineering, you have mathematics. There are so many fields that are coming together, into what we now know as a fusion of what we now know as bioinformatics. So maybe going forward in the next 20 years from now, maybe bioinformatics may even break down further, and then we can begin to have other aspects. But for now, we all call it bioinformatics, but it's a very big field. And whenever you see bioinformaticians discussing, you find that many of us are actually at different points of application. Actually, some people are doing medical imaging, some people are doing data storage and retrieval, some people are designing tools and software and anything in web development, software development. That's where some people are. Some people are actually looking at translational research, where they are looking at how to apply to medical field and do what we call precision medicine. So there are many of us coming from different fields, and there are different research areas and research topics really that you can find when you talk about bioinformatics as a whole. So also as a form of also defining stuff, I know every one of us here, we are not new to what artificial intelligence means, but basically it's something that has become part of our everyday life. And I think for the most part, it's really difficult now for us to do without artificial intelligence going forward. And I'm sure that, in the next 20 years, our kids will start to ask us questions how, we truly survived doing stuff without using any form of AI at all. So you find people who, right now, I'm sure if you are going to Lagos, you want to check on the map to see where the traffic is, to see what the best route is. Many of us also use it for checking weather predictions. So there are so many, applications of AI that have become like our day-to-day life, and they are so part of us. For example, those of you who use chatting systems or WhatsApp, for example, you see that for your own WhatsApp, it's been customized to the kind of words or the kinds of names or the kinds of register that you use daily. So for example, my children's names are already known by my own WhatsApp to the point that I'm typing the first two letters and the remaining letters are automatically given to me. because it knows their names already. Of course, in your own WhatsApp, the register there will be different from mine. So you can see how WhatsApp is able to use the power of AI to be able to learn the words that you use every day and automatically add them, to your own language, or, what I call a register, by the way. So artificial intelligence has come to stay, and it's something that people have started to apply even in all fields of life. So the medical field is not uh, any less in terms of applying it. And that's where, bioinformatics, or that's where anybody who is looking into biomedical research would start to think about how can we bring in all these methodologies that are already existing, and how can we bring them, to bear and make something beautiful out of it. So the keyword I'm trying to look out here is the what technologies. So some of you guys think about AI as just one big system, but it's a set of technologies really. And what I'm trying to do today is just to break down everything and see which aspect you're familiar with and you're interested in and you like to explore further. So this picture basically just shows us like a general overview of AI, and the different aspects. So there are different technologies in AI, and one of the subset of that is machine learning, which is where I am going to be focusing on today. But there are different aspects of AI. So we have robotics, we have computer vision, we have knowledge representation, which you can also call expert systems. We have speech recognition, which you also call NLP, which is natural language processing. Some people call it text mining. Okay, so depending on any field that you want, there's data mining. There are different aspects of it, but one of the subset of AI is machine learning, and that's where I really want to dwell on today. So machine learning basically is just looking at how we use algorithms, and I want to believe that we know what an algorithm is. So we use computer algorithms basically to learn from data. So basically, machine learning cannot work without data sets. So we have data, and we are trying to learn patterns from that data. And we use it to predict, we use it for optimization, we use it for detection, so many things. And somewhere down there in the middle here, let me see if my pointer is going to work. Yeah, so somewhere here you will see some popular machine learning algorithms, okay, random forest, SVM, regression trees, neural networks, autoencoders, deep learning. So deep learning is a kind of another further subset of machine learning. And inside that deep learning is where you now get to find your generative AI, and those ones seem to have been more popular. But we have been using machine learning and deep learning before, generative AI models, sort of took over, and that's what everybody is so fascinated, about. So machine learning, I've tried to describe it. I don't want to go over it too much, but basically what we're just doing is to develop algorithms that can automatically learn patterns and relationships, and we can use that to predict the future, or we can use it to predict unknown data. So there are different types of learning. We have supervised learning, we have unsupervised, and we have reinforcement learning. So without going into too much detail, in supervised learning, the data set is what is different. So in data set, in supervised learning, data set is labeled. In unsupervised learning, the data set you have is unlabeled. So it is an unstructured data, and then we just try to find what is the pattern or what's the structure in this data. In reinforcement learning, you are trying to teach the model by compensating it when it makes a good decision, or punishing it when it does not make a good decision. So gradually over rounds of training, the model is able to know what the good path is and is able to follow it. There's another subset which people are beginning to look at now, which is called semi-supervised. And semi-supervised is somewhere in between supervised and unsupervised, where you have a large set, some parts of it have labels, and then we use those ones that are labeled to train the classifier, and then we now use that one that has been labeled and train the model to then predict for the unlabeled data. So basically that's semi-supervised. So if you look at this picture, it also tries to explain better, what supervised, unsupervised, and reinforcement learning is. But I'm not going to go too much into detail, but just a way to just take you step by step. So why are we talking about machine learning? Why are we looking at how why it's useful? one of the things is that over the years, like when I started bioinformatics in 2007 was when I can say that was doing my master's, when I had, my first hands-on projects to do in bioinformatics. We're mostly using statistical methods. So things like t-test, things like ANOVA, which is analysis of variance, standard deviation, regression, hidden Markov models. There are plenty. Bayesian networks, for example. So there are so many models. For example, during my PhD, I used a Gaussian mixed model method that we had used. But right now, there are so many machine learning methods that are now, showing powerful results or even better prediction, much more than the statistical methods. So I would say that when bioinformatics started, we're heavily using statistical methods. And if you still see till tomorrow, you will still see heavily some of these statistical methods still being, projected, basically. But now machine learning is now coming in and it's now sort of taking over. So I remember when I was defending my PhD in 2017, it was the first time that I saw one machine learning model that they had used for gene regulation. And I was like, wow, this is interesting. And then immediately I finished, I just was very happy to quickly start investigating. At that time, it was still very new. But today, for gene regulation alone, I'm sure that we have more than 50 methods that have been developed for predicting gene regulation. But at the time that I started, most of the tools that were in application then were mostly statistical. So, like I said, I used the Gaussian mixed model method, but which was supposed to be an improvement over the Bayesian method. So, statistical methods in bioinformatics are very popular. And like those of you who are in bioinformatics, you see that most of the methods are in R. R is a statistical software. So that will also tell you how, how the development of bioinformatics. But now we are moving gradually and heavily towards Python. So if you see now, Python has become almost heavily used many times over and again. And that's because machine learning is beginning to gain ground, and we see that it has got a lot of strengths over statistical methods. So from my own little observation, these are the few advantages that I see that machine learning or AI is having over statistical method. Number one is in handling complexity. So many of the statistical methods can handle mostly linear relationship. Y is equal to mx plus c. Okay, looks like a simple linear regression model. And then they try to add a little bit of, complications here and there, but it's still not strong enough to handle non-linear relationships. Another thing is about scalability. I remember during my PhD time when I was doing a gene regulation data set, at the time I was working with microarray data, and then eventually RNA-seq data was introduced and I had to move and update my pipeline. I remember that the method really struggled because the range of values you get in an RNA-seq data set is far larger than microarray. So eventually we were able to get it done, but we still saw that the method or the traditional statistical methods were struggling with large data. But machine learning is, in fact, when it comes into millions of observations, millions of features, that's what machine learning wants. In fact, it's enjoying seeing large data sets. So machine learning has that edge over statistical methods when it comes to dealing with large data sets. And as we see with new technology, coming up every day, even the biotechnology that they are already improving day by day, you will see that the dimensions of data that we are getting are in millions. And you could even see when it comes to developing new, what's it called, storage infrastructure, just like what the third gen is bringing on board to be able to help us to be able to handle these large data sets. So we cannot remain at this level of having only statistical methods. So the other interesting part of it also is the feature selection and the feature engineering approach, which I believe some of us may know. So with feature selection, we are able to remove redundant features. What do I mean? So like you have a set of observations and you have the label. But the truth is that not every one of those features will contribute to your label in the end. So what you want to do is to remove all those ones that are not, informative enough, and to just work with the ones that are informative. So basically, when it also comes to prediction accuracy, you find higher performance basically with machine learning algorithms. And so also when it also comes to adaptability to new data set. So I have put in references here, and I hope that, if I'm not able to explain enough, you can actually go and check again these papers, and you'll be able to find more explanation. So in the next part of the second half of my presentation, I've already spent 15. In the second half, I'm going to highlight each of the different application areas of AI, or invariably machine learning now in biomedical research, okay, one after the other. So the first one I want to talk about today is genotype to phenotype prediction. So without using too many biological terms, what do I mean by genotype? So genotype basically just explains the entire genetic information for a particular organism. That's what we mean by the genotype. And the phenotype is like the physical trait that you are investigating. So let's say, for example, I am investigating an infectious disease, maybe malaria or COVID-19, for example, and I could decide, okay, my phenotype is people that are infected or people that are not infected. Or I could pick my phenotype to say, my phenotype of interest is that maybe I'm looking at different classifications, maybe people that have severe malaria, or people that have mild malaria, people that have, let's say, asymptomatic malaria, or people that have cerebral malaria. So you can group them maybe into like 5 groups, and those that are negative are those that are not having the disease. So when we say phenotype, we are just referring to the traits, the physical traits that you are investigating, or the observable trait or the characteristic that you are investigating at that time. Then if I say genotype, I'm referring to the entire genetic composition of that organism. So it could be the set of genes. Sometimes we also use transcriptomic data sets where we're looking at the gene expression data. But many times we use the entire genome, the genome data for that organism. There's also what we call GWAS. That GWAS is genome-wide association study, where they are only looking at the genetic variation. So for example, let's say we have like a whole genome of an organism. That genome is, let's say, 3 million letters. So with GWAS, what that GWAS does is that because we are 99% similar, everybody you see, human being A, human being B, human being 10, we have about 99% similarity, and there's just 1% of difference amongst us. So in that 1% now, it's going to check for where the differences are and just say, okay, this position for everybody is a G, that is the nucleotide at that position. If somebody has an A, then it's just record that this is A for this position. So GWAS only picks where the differences are in the genotype, in the whole genome of that organism, and say this person, instead of everybody having a G, this person has an A, and that is the difference. So it will only record the single point mutations or the single point variations. So we have a data set like that. It's a bit less heavy than dealing with the entire genome of that organism. But what am I trying to say? There's what we call genotype to phenotype prediction, where we have a particular phenotype. Say we are dealing with height, for example, and over the years, it has already been established that height is genetic. So if you are short, there's a heavy contribution of your genome to that height. So we now want to find out what are the genetic differences that are contributing to the differences in the height. So we could now pick, height is a quantitative, is a continuous trait. Okay, so we can pick different people with different heights. And now maybe we can group those that are very tall or those that are very short, depending on how we want to do the study. But when we do genotype to phenotype prediction, we are looking at where in your genome do you have a difference, and how is that difference contributing to the characteristic that we see. Another example, I want to deal with hair. I could pick ladies that have naturally long hair, and I could pick ladies that have naturally short hair. And then we check, is there anything in their genetics that is contributing, to the long hair that we are seeing? They have used this trait extensively in plant studies, where there are some desirable traits in the plant. So for example, you have watermelon that is very sweet, and watermelon that is not sweet. And then they check what in the genome, what variation, what different, when you say variation, means there's a change in that person's genome. So what variation is in this watermelon that is making it to be sweet, that is different from this one that is not sweet. Or maybe there's ugu leaf from the east that people know that Igbo ugu is very thick and it's like very desirable, whatever. So maybe there are some desirable traits. So they try to pinpoint the specific part of the genome that is giving that desired trait. And then once they identify that, they can now engineer that, bioengineering, and that's what is leading to having GMOs. So when you hear genetically modified organism, genetically modified food, that's all that is happening. Like you have a desired trait, you're able to locate or pinpoint which genetic variation is making that to happen. And then they now make that genetic variation artificially to create that trait. So we see that a lot in plant studies. But for human studies, we like to use it to diagnose. So for example, in cancer studies, they have done it extensively where they are finding who are the people that have certain variations, and those variations have been known to associate with certain diseases or certain traits whatsoever. And then we can also use it for, designing therapeutic interventions. So let me not go too much into the application, but because we are technical people here, these are the different methods, and I've put here a lot of links to papers that you can read that tell you how you can use your known machine learning methods to combine, to go from genotype to phenotype. That means you have a set of genotypes and you are able to pick which genotypes are responsible for the phenotype. I'll give you another practical example. So one of the ones that one of my students had studied was to look at drug response. I'm sorry, I may take a little bit of time, but let me just explain these things very well. So we're looking at drug response for different Plasmodium falciparum strains. So what they did was that they picked people that were infected with malaria, and they took their blood sample and they tried to culture the parasite in the blood. And the next thing they did was try to treat these parasites from that person's blood with different drugs. So of course, because there's drug resistance, some the parasites for some people's blood, sometimes the parasite died, sometimes the parasite did not die. So what they then did was to give a label, parasite A. Let's say they had 100 patients. Please let me pause. Can you see me? Can you hear me? I hope everything is fine.
Yeah, you're good.
Thank you. So they took, let's say 100 patients, and then they tried to find people that the parasites that responded to the drug and the parasite did not respond. So the ones that did not respond, they labeled them as resistant. The ones that responded, they labeled them as sensitive. Now the second step was to pick the parasite and go and sequence them. So they took them to the sequencer. Of course, sequencing is expensive, but now I'm happy that we have a sequencer in Covenant University. So with a sequencer, we are able to generate that genome I was talking about. So we had the entire genome of Plasmodium and the label of the ones that, so each of those parasite strains, we knew their genome and we knew the ones that were sensitive and the ones that were resistant. The next thing was, let's use machine learning, because there's no way we can use our natural eyes to just be scrolling through millions of letters. If you look at what a whole genome sequence is, you're just seeing ATCG, thousands and millions, and you can't do anything with your natural eyes. So we then, of course, so many people had done so many research in that area. We just picked one model and we enhanced it. Unfortunately, we are still not able to generate our own data set, but I'm hoping that, with funding from CAPICASE, we can start to generate our own indigenous data. But this study was done in Kenya, and then we're able to redo that study with better, machine learning, and got a better performance. But data set was generated in Kenya. But basically, what that can help us to do is to find what genetic composition is making these parasites not to respond. And we found that for the different drugs, because they used about 10 different malaria drugs, we found that for the different drugs, there were different genetic variations that were responsible for each of the drug resistance that they had seen in that data set. So we can do something like that, in our own environment, and try to put up like a genetic or genomic surveillance system where we are monitoring how the parasites are getting mutations, and we can quickly say, oh, this is the variation that we are seeing, and this is how fast that variation is spreading. Because if it spreads so fast, then it means that that drug eventually over time will become useless and it will not work anymore. So that's one simple explanation, but it's not as simple because you have to deal with a lot of large data sets. But these are some samples where you can see. And if you are interested, you want to try this out, you will find some sample genomes online. I know that for human genetics, you may not find human genetics that is data set that is open. They don't really release human data sets easily, but you can do with microorganisms. So if you are doing parasites or bacteria, you can easily find all those whole genome data sets for some of these lower organisms easily without much ethical approval. But if you want to work with human genetics, which we will find, which will be better for us, but is that we are generating by ourselves or we have to go through long process to get data sets. So this is a very interesting application of machine learning. And I've also tried to put another case study here where they try to use machine learning, deep learning in particular, to identify the variations that were responsible for, a particular disease. So here we have different diseases. We have type 1 diabetes, we have type 2 diabetes, and we have hypertension. And they were able to identify different variations in different participants or patients in that study that were contributing to that disease. The next one is biomarker identification. So somebody will ask me, what's a biomarker? So a biomarker basically is like, it's a protein, and that protein is an indicator of something. So let's say, for example, if you know how this pregnancy test works, I'm sorry I'm using something that is gender specific, but if you have ever seen a pregnancy test, like before you get urine, you dip it inside the urine, and it's able to tell whether you are pregnant or not. Have you ever wondered how it works? So basically, the idea is after some point in pregnancy, you begin to have some proteins that would show either in your blood or in your urine. But many times at the early stage, we like to use a blood test to specify. But maybe by the third week in your urine, you start to see those proteins showing. So that protein is a biomarker because it is indicating the presence, so it is signaling the presence of something. So we use these biomarkers a lot for detection. And if you have seen malaria, there's this malaria RDT now. They use it in Medicare a lot. So what they just do is to take your blood, put it on that diagnostic test kit, and then it can tell you whether you have malaria or not. How does it work? It's just checking the proteins that have been expressed. So if you have the parasite in your blood, the parasite will express its own proteins. Okay, so if that protein is there, it signals that it is there. The same thing for COVID. If you have had COVID, the virus, there are some, either you're using the virus proteins or you're using your own immune response that actually shows that you were infected by either a virus or a bacteria or a fungi, depending on whatever pathogen you were infected with. So biomarkers are like, they have been extensively studied, and machine learning really now is being used to actually identify biomarkers. Detecting biomarkers in the lab can take a lot of time. And so machine learning now is becoming very useful. These are some very popular methods, and here are also links to articles that you can read on, applications of machine learning to detecting biomarkers. So for a case study, this is one particular study that shows how random forest and SVM was used as a classifier to be able to identify, a biomarker. So I'm sorry, I'm not going into too much detail of specifically how, but let me just I want to just achieve that overview today where we're able to see all that machine learning can actually be able to help us to do. So here is multi-omics, and I particularly enjoy this because I've also done this with one of my master's students. So this is multi-omics. What it means is we have a disease and we are studying that disease from different sites. So there's metabolomics, there's proteomics, there's transcriptomics, there's
Hello, ma.
epigenomics. I can hear you. Were you saying something?
Hello, can you hear me?
Yes.
Okay, so I said you have 5 minutes.
It's echoing. Let me put on my is it my own car? Please go ahead. I didn't hear you.
hear me now?
Yes.
Okay, so 5 minutes. You have 5 minutes more.
Thank you. I'm almost done. So for multi-omics integration, we just try to integrate different omics, and then we try to integrate data sets and analyze it as one complete data. So you also have links to we also have links to what the multi-omics just a minute to my system is freezing. Okay. Yes, so these are all different methods also that have been used for data integration. The reason why this is interesting is because you have different types of data sets. And the problem here is that you want to integrate them into one so that you can now analyze them as one whole stuff. Next one is drug repurposing and drug target identification, which is very common. But one of the things that I found this interesting is how we can use existing drugs to treat new diseases. I remember how when COVID came. Okay, so this can be very useful for preparing for new pandemics, because eventually, as it is now, pandemics are part of life and they may come. So finding how we can repurpose existing drugs in readiness or for treatment of future pandemics is actually something that is very interesting. So next is biomedical imaging. Neuroimaging is very popular because, of course, anything that has to do with the brain needs a lot of images. So if you are talking at anything psychiatric or whatever, you need a lot of neuroimaging. But there's not only neuroimaging, there are lots of biomedical imaging technologies that we have. And we need machine learning. We now use machine learning to be able to analyze them. One of what we have tried to do with the microscopy training was we are trying to see how we can bring in machine learning into analyzing images from the microscope. So the microbiology students only just use the microscope, generate their images, and then they do their diagnosis and go. But we are trying to also see how we can develop our own automated diagnosis system, where from the microscope, we can design software that can automatically, detect the image and do the diagnosis. That means when there is no expert, we don't have a pathologist, we don't have a microbiologist, but we have our microscope. We can now connect, a software that has been developed to automatically look through the microscope and then do the detection. Or we can upload the images from the microscope into a software system where we can now begin to automatically detect or diagnose the presence of any pathogen. So the same thing goes for other imaging, MRI, CT scans, all manner of imaging, whatever detection you want to do. For example, we've seen where people have also detected COVID from chest X-ray, all manner of things. So several softwares that actually deal with images. Of course, CNN is still one of the best methods, for anything image analysis. So the last application is on electronic health records. And this one is basically just helping us to be able to manage and maintain our digital repository. In Africa, these things are still like stories. But in other places in the world, these have been well established. And when you have well-established health records, we can now begin to deploy our machine learning tools where it's able to now predict from just the health records alone. So I'm not talking about system to develop the health record. I'm assuming that the health records exist. Now, there are several machine learning tools that can now, be developed over the health record system to be able to predict. One very good example that I like to use is this predict prostate tool. And I've put the link there. It was developed in the UK using the NHS. I'm sure you know what the NHS is. So the NHS has massive amount of data sets. And what they did was to just pick the men that had prostate cancer. And they tried to use their patient characteristics. So the age, the treatment, different clinical factors, not genetical, just clinical, the patient characteristics was what was used in the prediction system to be able to predict at what age these men will develop prostate cancer. Now, if you go and Google this predict prostate, they have used this method in several European countries where they also have a very detailed health record system. So for many people in the abroad, when they are born, or when they are born to maybe when they die, you can easily trace their, their record. And with that, they are able to use, at least this predict process is one very good example. In fact, it has nothing to do with genetic testing at all. It's just going through their health record and their clinical record system and their treatment history and able to predict whether they will have prostate cancer. And there are so many other diseases which some other people have tried. So because of time, those are the few that I want to say, but there are many challenges, when it comes to applying AI, machine learning. One very, very big one that is a challenge is imbalanced data set. Many times, the data sets that come, to us are like in different. So, for example, let's say you have a study that has 4 classes, for example. Many times, most of the data sets are not properly portioned across the classes. So you may have to now, either you have to use small to undersample the overrepresented class, or you oversample, whatever you have to do. But sometimes, you may not give you the perfect performance that you're expecting. There are so many challenges. Hyperparameter tuning is another thing. From one data set to another data set, you always have to, correct for the hyperparameters that will best fit, that data. And there are so many issues that I can talk about from now to tomorrow. But I just hope that I've been able to give you like an overview of how AI really can be used in biomedical research. So these are some selected references, and I'll be happy to take your questions. Thank you for listening.
Answering session now. So online participants, please, if you have any question for Dr. Itunuoluwa, please wave your hand so I can call you. But before online participants, we have physical participants here. Does anyone have any question for Dr. Itunu? By the way, that was a very, very great presentation, Ma. Thank you, thank you, thank you. Okay, so question?
Do we have any question for her here? Okay, Mr. Odeme.
Doctor, thank you so much for very insightful presentation. My question would be on the sequencing part, where you talked about, you mentioned in passing that fortunately we have now a sequencing machine in Covenant University. And I'm a bit concerned on whether or not we are able to use. Can you shed more light on what it would take for CAPICASE to put that to use, and for us to really have data that we have from the samples that we have? What does it take? Why is it looking impossible for us to have or replicate what has been done in some other universities outside? I don't know if my question is clear, Ma.
Dr. Ma, do you want to
Harvest all the questions and respond to them once, or you will respond?
Yeah, so because of time, can you hear me?
Hear you.
Yes. So because of time, I'm hoping that I can answer everything at once. So I will let other people also make their presentation.
Hello, Ma, we can't hear you.
It's like you're muted.
Yes, I'm unmuted now. Can you hear me now? Hello?
Hello, Ma.
Yes. Can you hear me now?
Please.
Hello?
Hold on, Ma, we can't hear you.
No, it's not that. It's not that.
Okay. Comfort, can you hear me?
Yeah, I can hear you, Prof.
We can hear you.
Yes. Okay. Dr. Itunu want to harvest all the questions so that she will just respond to them at once. That was what she said. I am hearing her clearly from here. So I guess you need to check your maybe link at that end. So if there are other questions, she's willing to harvest all the questions, then she will respond to them at once. By the way, well done, Doc. That was beautiful.
Thank you, sir.
Coherent. Okay. So can we, if there are other questions? Okay.
Okay, so while they are trying, I think
Question, do you have any question for her?
let me start answering because of time. So to the person who asked about the sequencer,
We have one
Online participant.
I've seen it.
He was asking about, let me see, I think I saw a chart. Okay, from Messi now.
So she's asking, Ma, as a biologist, who would want to integrate machine learning into her work? What training will you advise one take? Okay, so she's saying as a biologist that wants to try and integrate machine learning into her research, what type of training will you advise her to take?
Okay,
Miss Messi.
thank
I
you. So
believe that that's
let
your
me start.
question.
Yes, thank you. Let me start responding to them so that we can wrap up. I believe that there are other presenters for the week. So I'll start with the first question on the sequencer. So the sequencer is in the cancer genomics lab, and Professor Rotimi is the one sort of spearheading that initiative. So I'm not sure to the extent where we are, but I know that they had to set up energy whatever installations for the sequencer so that it doesn't get bad, and that they also had to connect it to like an analysis platform, so that once the data set is coming, it's going straight. So I'm not exactly able to tell you this is where we are, but I know that a lot of effort has been going into establishing that sequencing lab, and I know that a lot of money also has gone into it. So let's just wait and hope that eventually, when it's fully functional, the CAPICS community will be able to get a presentation, hopefully. But I know that last time I had a conversation with Professor Rotimi on this, he was saying that they have already enough samples for that sequencer's lifetime. So we may not get to use it, but what it tells us is that there is hope, because I know what it takes to actually get the sequencer to work. So if you can work with us, hopefully the malaria group also, we're also planning to also get our own sequencer in the end. So, but for the cancer genomics lab, they have more than enough samples already for the sequencer to even start working on. And from what I was told, the sequencers don't last forever. So after they have used them for maybe about 1,000 or 2,000 samples, they go bad, and then you have to get another one. So I don't want to speak too much about what I don't know much about. It's not my area. But if you want to know more information or get more information, please talk to Professor Solomon Rotimi. He can tell you more about it. Now, you talked about generating our own data, and that is very, very important. Most of what we have been doing has just been using online data, and there's a limit to how much you can publish that. So maybe now that the biological sciences are beginning to generate data, we can truly do what the bioinformatics aspect is. I didn't know bioinformatics lab should work in isolation. In a normal setting, we normally work in collaboration with, the biological sciences laboratories. So the idea is that once they set up their own pipeline and it's working fine, we receive the data for analysis. So I know that I've been speaking with Prof. Rotimi a lot about how to set up the data analysis pipeline, but they want to even finish the lab aspect first before they establish how the analysis pipeline and the variant calling pipeline will work. So eventually we'll work together in the end. And there are many things that I've actually been telling them about. I'm so happy that the microscopy one is finally, happening, and we are able to sort of synergize. But ideally, third gen and bioinformatics and the labs are supposed to all be synchronized. None of us should be working, like in silos. Unfortunately, that's what it is now, because everybody's still trying to build capacity. One of the things I told my student was, let's build capacity in anticipation for when these data sets will come, so that by that time we will have been very well equipped to handle whatever challenges or complications that they are. But in a real research setting, all of us should be working together. And that's where we hope to get to in Covenant University in about a year from now, at least minimum. We should be operating at that level, whereby the third gen lab is able to provide all the solution, and the bioinformatics people are also able to jump on the data and start to, quickly analyze and be developing analysis pipelines that can even run without us being there in the future. So the second question is about biologists training in research for machine learning research. So what I would say is, depending on what you want to do, there are different machine learning algorithms that you may need. So what you should ask yourself first of all is your research question. So your research question will then dictate what type of machine learning model you are going to use. So when you find your question, then you now go and read papers that are similar to what you have done. In fact, what I just tell people is start from the barest minimum. It's not really advisable to tell you to improve the existing model. So if you can even deploy the existing model, then for me as a biologist, you have succeeded. But if you are coming from the computing side, of course, I will ask you to go and look at what the challenges are with that model and try to give us a better performance. But at your level, I would say find a paper that is similar to what you have done. If there is maybe like a shallow machine learning model, which I feel may be easier for you to deploy on your machine, because one of the challenges also is computing resources. Many of these machine learning algorithms are heavy and they require a lot of computing power. So for the small laptop that you have, you may want to start with something like SVM, naive Bayes, KNN, simple artificial neural network, maybe like one layer MLP. You may want to just, start with this little, we call them shallow machine learning code because they don't require very heavy computing resource to run. So you may want to start with those. And if you can even maybe go on GitHub, look for a code that is working, if you know your way around, run them, put in your data and run, and just start from there. So I would not tell you this is the particular one to use. All the machine learning methods have their strengths and weaknesses depending on data sets that you're working with. But maybe you want to consult. If you consult, then we may be able to give you better advice. And then for the chat, there's a recommendation on ethics discussion about AI. Yes, I agree, and I was not able to, include that in the presentation. I've taken note of that for future presentations. Thank you. I hope I've covered all the questions. I'll be happy to. Okay, yeah, I think that's it. Thank you very, very much. I'll be in the chat if there are other questions.
Hello, Ma. There is one question from Dr. Taufik.
He said, have you ever handled a case where a sample belongs to more than one label classes? How did you go about that?
Yes, so I'm sorry I missed that one. Okay, so usually when expect if it's just supposed to be yes or no question, and or maybe there are multiple classes, if normally, for example, let's use the case of malaria that I gave, I don't expect one person to have both severe malaria and mild malaria at the same time. So if you are seeing that case, then that means that is a case of misclassification. Many times we remove those kind of data points from the data set because of course there's an error somewhere. And I didn't go into the step by step, but one of the things we do is to clean the data. So if you find such samples, we normally just remove them because we don't know what happened during the data collection process. That's my answer to that.
Thank you so much,
Ma, thank you. Dr. Taufik, I believe you're satisfied with her answer. And Miss Mercy.
Okay. So there is another person. Thank you, Ma, for detailed presentation. I think she's
Asking for the training she can to become.
So I saw that. I was already typing a response. So to start training, I would say that you can use several free online resources. So I recommend Data Camp, there's Coursera, there's Udemy. You can start from there. The first question is, are you, knowledgeable in programming? So if you don't know how to program, then maybe machine learning is not for you. So you may want to start to learn a programming language. I recommend Python a lot, but there are many other languages that you can use to learn to do machine learning. But Python looks like one of the most heavily supported languages. So if you don't even know how to program, maybe you want to start to learn about programming. And like I recommend to people, there are many textbooks or resources that you can pick. So after you have learned at least some basics in programming, how to move, so many times people don't even, today, ChatGPT is writing codes. Well, those codes are wrong. I've tried them, and many times you also need programming knowledge to be able to prompt it to give you the final answer that you're looking for. So you may just want to learn simple basis of programming and then be able to leverage generative AI to help you. But I don't think that generative AI will help you if you have zero knowledge of programming. So you may want to start learning programming, then maybe also learn a little bit of this AI and understand what machine learning